Thank you very much, everyone, for coming. We will start with our session now. Before I hand over to Professor Andres Floto to introduce our guest for today, I will just say that if you have questions during the talk, please just write them into the chat or mention that you have a question. And at the end of the talk, I will then call you up to ask your questions to our guest. If you want to take it away, Andres.
Great. Thank you, Andres. Welcome, everyone, to this latest edition of our distinguished CCAIM seminar series. My name is Andres Floto. I'm co-director of the Centre. And it's my great pleasure to introduce Professor Daniel Alexander, who is from University College London, and is director of the Centre for Medical Image Computing and deputy head of the Computer Science Department. Great. And today, Daniel's going to be talking about image quality transfer and democratization of MRI. So thank you again, Daniel, for speaking today, and over to you.
Great. Thank you for the invitation. Yes, so thanks again for the introduction. Very nice to be talking to you today. The topic I chose today is image quality transfer and democratization of MRI. Not sure how much that would mean to everybody, but I chose this because it's sort of the thing at the front of my mind at the moment. But I'll come back to what that means. I thought I'd just start actually with a bit of a wider view. I'll talk a little bit about where I come from and some of the other projects which actually are probably more associated with my name than this one. I'll just mention them briefly in passing, and then I'll get to the meat of the talk about the topic I wanted to focus on today. As Anders said, I'm the director of the Centre for Medical Image Computing at UCL. This is a centre that sits in the engineering school, primarily people from computer science and medical physics, but with various clinical people involved as well. We've been around for quite some time now, established in 2005, and the centre has grown steadily over that time. So it's quite a large centre these days, something like 25 associated faculty and around 200 or so researchers, students and postdocs and so on. And that critical mass gives us, gives us a capability to focus all the way across the imaging pipeline, from sort of very basic engineering advances in imaging techniques, devices and sensors and so on, kind of going around this circle through to people doing modeling, image reconstruction, and then various image analysis groups, disease modeling, through to kind of frontline clinical systems. And so, really we've become one of the primary conduits for new ideas emerging in engineering and computer science through to application in the clinical research in the medical faculties at UCL and in clinical practice at the various hospitals associated with UCL. So that's what we do. This just kind of summarizes what the expertise we claim on the left. So that reflects, us belonging primarily to computer science and medical physics. And really the kind of applications on the vertical bars here. The point I often make about this slide is that we don't have a particular medical focus on any medical speciality. Probably the biggest four applications for us are the first four here: neurology, oncology, surgery, and respiratory, but by no means do we limit ourselves to those topics, and we certainly have smaller activities in a range of other application areas. And I think actually this kind of sort of specialty agnostic approach is part of the power of an engineering-based centre in medicine, because one of the things, the key features I think of a centre like CMIC is, and probably similarly for CCAIM, is that it enables us to spot opportunities easily to transfer ideas that have maybe made an impact in some way in one medical specialty and repurpose them for application in another. And with that theme in mind, I'll just talk about a couple of topics that I think kind of quite nicely exemplify that. I mean, these are topics of my own research, and the same is true of other activities within CMIC, but just to highlight that capability. So the first topic I'll mention is the idea of microstructure imaging. This is a model-based imaging paradigm where, sorry, I'm just going to minimize that. Okay, thank you. Model-based imaging paradigm where we try to quantify and map microstructural features of tissue over an image. And my work on this dates back quite a number of years now. I'm going to give you a very brief summary of it, but if you want some more information on it, there's a recent review article that's kind of explains not just my work, but the state of the field as it was at least about 4 or 5 years ago now. Struggling to change slides here. There we go. Okay. But yeah, so the microstructure imaging is a quantitative MRI technique, often using diffusion imaging, but not limited to that. The aim is to produce quantitative maps of quantitative tissue parameters. Things like these maps here in the first row. So these are features of brain cells here. This map here is the density of brain fibers within the brain, and sort of maps that, makes an actual measurement of that over each voxel of the brain. And similarly, this is a map of orientation dispersion, how so there can be millions of these fibers within each image voxel. And this map shows in each voxel a measure of how dispersed the orientations of those fibers are. So for us, the journey in microstructure imaging started in neuroimaging with techniques like NODI, which is where these maps come from, and is now a sort of quite a widely used technique in neuroimaging, part of sort of various standard imaging protocols. So yeah, producing these kind of quantitative maps, orientation dispersion, neurite density, and this third one here is a kind of a confounder really that you have to estimate to estimate these maps. It's a kind of partial volume with fluid in the brain. So the technique works by designing a mathematical model that links these kinds of biological quantity to the MRI signal, and then fitting that mathematical model to a set of measurements in every voxel of a multi-channel MR acquisition, which has been tuned specifically for sensitivity to those particular parameters. Modern versions of this idea are quite AI heavy, both in the model construction, often derived from quite complex simulations now, where we use regression networks to map those complex simulations to MR signals, and in the estimation, you can do very rapid model parameter estimation by learning those mappings a priori. And even in the design of the experiment itself, often those rely on modern AI techniques as well. So yeah, as I say, the journey started in neuroimaging, but then having this kind of critical mass of applications in different areas allowed us to repurpose that idea in different contexts. So one which is showing nicely, kind of leading towards impact these days is to repurpose to look at solid tumors, and that's the idea of the verdict technique. So initially kind of put forward in 2014, but then showed a promising proof of concept in looking at prostate cancer in 2015. And so really the aim here is to try and replace what is the standard of clinical care in prostate cancer at the moment, which is a prostate biopsy. It's as unpleasant as it sounds, a prostate biopsy, and so that the potential of replacing what is a highly invasive procedure to test whether a cancer lesion in the prostate is malignant is very beneficial if you could use an entirely non-invasive MRI technique. So that's the aim. The timescale here is quite interesting. From first proof of concept in 2015, we've then been through various layers of clinical trial to establish the real-world benefit of of verdict. The aim being to demonstrate that it will discriminate a clinically significant cancer from an insignificant cancer, which sort of standard MRI techniques can't do, which is why we still rely on the biopsy. But having been through a couple of trials now, the latest one actually has a pretty strong conclusion. This was a retrospective trial where people having the verdict technique also had the prostate biopsy, but the conclusion was that 91% of those people, the same decision would have been made using the non-invasive technique as the invasive biopsy at a relatively small cost of in terms of false negatives. So that, it's quite close now to being able to deploy that, at least locally, in clinical practice. And yeah, similarly repurposing this for other areas. We've been looking recently at the placenta, for example, where similar ideas, this kind of model-based imaging paradigm provides useful markers, early indicators of pregnancy problems. So that was kind of topic number one. Second one I wanted to mention was the idea of disease progression modeling. So the broad aim in disease progression modeling is to try to recover from sort of sparsely sampled and heterogeneous patient data sets, longitudinal pictures of how a chronic disease evolves over time. My line of work on this was really inspired by a debate that erupted in the neurology community, probably 12, 13 years ago now, about the sequence of events that a typical patient with Alzheimer's disease experiences during their journey. So that debate was really fueled by the emergence of large multimodal patient data sets at the time, and allowed people to sort of infer that the earliest kind of signs, measurable signs, were an imbalance of proteins in the brain, which you can see with things like CSF lumbar puncture or PET imaging. And then the kind of deposition of those proteins in the brain gradually kills off brain cells, which eventually is observable as morphological changes in the brain, shrinkage of various grey matter regions, for example, which you can see using structural MRI. That loss of brain cells eventually leads to reduction of cognitive performance, which you can measure using various cognitive tests, and ultimately to kind of widespread personality changes and loss of function. So, various of these hypothetical models emerged at the time, but none of them was really directly informed by the patient data sets. And so, various computational people like me set out to try and construct algorithms which could learn these pictures directly from those the data that were available at the time. It kind of sounds simple on the face of it. If you simply take a set of measurements from a bunch of people at early stages and then follow them closely through the course of this disease, you could reconstruct a picture like this quite simply. It's not as simple as that. Firstly, because it's very difficult to find people who are on that disease journey at these early stages, because they appear perfectly normal. Even if you could find a decent cohort of people at these early stages, it's not really practical to follow them, making temporally dense measurements over a period. This could be 20 to 30 years. It's not practical to measure them repeatedly over that period of time. So those kind of longitudinal data sets just simply don't exist. But what we do have is large sets of kind of snapshot measurements from individuals passing through research studies or dementia clinics at small number, often just one time point, and at an unknown position along this disease timeline. So the modeling and learning challenge then becomes trying to piece together a picture like this from a set of snapshots from different individuals at unknown positions along this timeline. And that's the basic challenge in disease progression modeling. So, various solutions to that problem arose around, in sort of shortly after those hypothetical models in the sort of early half of the last decade. And I was part of a European project which really focused on that, bringing together various techniques and rolling them out from looking, first of all, at Alzheimer's disease and then a range of other neurological conditions. I don't want to focus on that too much. What I want to focus on is there's some more recent advance in that technology, which is the idea of sustain or subtype and stage inference. So the sustain algorithm relaxes one key assumption that the early models all made about in order to reconstruct these disease timelines. And that assumption was that everybody is on basically the same trajectory. They're not, particularly in, in most chronic diseases, there's quite broad heterogeneity of the trajectory that individuals experience. And SUSTAIN sets out specifically to identify a set of subtypes of a disease, each defined by that trajectory, the trajectory of change that individuals within that subgroup experience. So it's essentially an unsupervised learning algorithm that simultaneously identifies a set of disease subtypes, the trajectory that defines each of those subtypes, and an assignment of individuals to both subtype and kind of position along that trajectory or stage. I won't go into the details, but just again to sort of highlight that this, although it's a technique that was specifically designed for understanding Alzheimer's disease better, since constructing that algorithm, it's found application in a much wider range of medical areas. First of all, rolling out to other kinds of dementia, other neurodegenerative conditions. This is an example of multiple sclerosis, and even beyond that, to look at other chronic diseases like this is chronic obstructive pulmonary disorder, respiratory disorder, but it's also been applied in osteoporosis and ophthalmology conditions as well. Okay, so that was my lead in. Now to the main topic I wanted to talk about, which is image quality transfer, and the context of that is this idea of democratization of MRI. So let me take a zoom out to kind of put that last statement, second half of my title, in context. What I mean by democratization of MRI is making it available to everybody, not just those of us that are fortunate enough to be in kind of comfortable high-income countries with lots of medical equipment available, but much more broadly across the world. This is a map of, let's think about Africa to start with. This is a map of Africa constructed by Aduna Anozodo here as part of this paper that's just been accepted to NMR Biomedicine. And what the map shows is the accessibility of MRI across the African continent. The color bar is showing the number of MRI scanners per million people. And what we very quickly noticed from this figure is that particularly in this sort of sub-Saharan region of Africa, that availability is very, very low. And typically it's somewhere between 0 and 1 MRI scanners per million people. And you can contrast that with the figure in say Western Europe, where that figure is well in excess of 10 scanners per million people. And even further than that, the scanners that are available are typically kind of 1980s technology, much older machines, less powerful machines than those that we are that are commonplace here in higher income countries. Part of the reason for that is a part of its cost, but it's not only cost. Actually, probably a bigger factor is the available infrastructure. So modern MRI scanners do not cope well with power outages, which are common in many of sub-Saharan African countries, whereas some of the older technologies have a fixed permanent magnet that is much more robust to a power outage. So often the technologies are more driven, the technologies in use are more driven by the infrastructure than the cost, although it's a bit of both. So how can we go about sort of trying to increase that availability? Well, the challenges are pretty profound. As I say, part of it is financial. These machines are expensive, not just to buy, but also to maintain. And in fact, possibly the maintenance costs are a bigger barrier than the actual purchase of the machine in the first place. And then there are the infrastructural challenges, which is that a modern machine requires a steady power supply, which we don't have. So you have to then put in more money to include a generator or something that will stabilize the power supply. So those are certainly challenges, but probably an even bigger challenge is just the sort of institutional educational setup in many of these countries currently. There simply aren't the people available who can use these machines effectively, even when they are placed there. There's a kind of chicken and egg problem. Why would you educate people to be experts in a machine that isn't available? And why would you provide a machine to make it available when there aren't the people who know how to use it? So yeah, there's a chicken and egg problem that sort of prevents at least, you know, rapid deployment of the technologies that we are used to, even given the funding, if it were there. However, I think some of the recent movements in the MRI community provide some traction on this problem. So there's a notion recently of the idea of accessible MRI, and part of the idea of accessible MRI is finance, making machines that are cheaper to buy, to install and maintain. But there's an additional aspect to that, which is about the usage of the machines, just making them more push button and requiring less expertise. So, you know, in particular, not requiring a highly trained MR physicist to be on site 24/7 to, you know, maintain the machine and make sure it's doing the right thing. So here, you know, slides just showing some examples of the kinds of technologies that have appeared just over the last couple of years. Top left, this is the Freemax machine that Siemens, one of the big MR manufacturers, introduced just a couple of years ago now. Unlike most modern MRI scanners, which are 1.5 Tesla or 3 Tesla magnets, this is a lower field strength. It's 0.5 Tesla. It has a much smaller footprint, so it's a cheaper machine, easier to install and easier to ship. The magnet has a much lower helium content than most modern magnets, so it's cheaper, and it doesn't have such a problem with intermittent power supply. But it is still, you know, an MRI scanner of the traditional form. It's fixed. You have to take the patient to the scanner and you put them inside the, you know, the big tube here, like most MRI scanners. There is a, you know, perhaps a bigger departure is the emergence of smaller portable MRI machines, and that's what the other two pictures I'm showing here are. So the first picture here is a machine developed, sort of introduced into the market by the company called Hyperfine, also a couple of years ago now. You can see that it has wheels. You can wheel the thing up and down a ward, so you can take the scanner to the patient rather than vice versa. Much more like a, you know, it looks really like an ultrasound machine. It's that kind of size and shape, and, you know, like an ultrasound, you take it to the patient. So this has been on the market, and there are quite some substantial efforts around the world to deploy these kinds of machine now to Africa and other lower middle income settings. This is another, there are other machines like this emerging now. This is another device, the brainchild primarily of Andrew Webb in this picture here from Leiden. And just a couple of weeks ago, I was in Nigeria at the same time that they were installing this machine in Uganda. And there were various other copies of this device being installed in a number of other similar locations. Recall that the Siemens machine was 0.5 Tesla as opposed to 1.5 and 3T, the typical MRI scanner, modern MRI scanners. These portable devices are an order of magnitude lower still in field strength, 0.05 Tesla, the Hyperfine machine, this one similar. And that comes with a significant downside. Plus side is portability and cheapness. The downside is that the images are much lower quality because of that lower field strength. So that's kind of where I come into this story with the notion of image quality transfer, which, you know, essentially is a method for using modern machine learning techniques to try to enhance the quality of images, particularly from these sort of low resource settings. So I'll talk about image quality transfer just for a bit now. I'm going to wind back the clock before I really started thinking about these low power MR systems to where I really started with image quality transfer, which was with a kind of a super scanner device, which called the Connecton. So back in 2013, maybe it was a bit later than that, 2013, 14, a bunch of us in the UK got together to seek some funding to persuade Siemens to build a super scanner, a Connecton scanner, It's actually just it's a fairly standard 3 Tesla MRI scanner, but the thing that's super about it is that it has much stronger magnetic field gradients. So although the field strength is about the same, the ability to make a gradient in that field strength is much stronger than usual. And that is important in, it allows you, particularly for certain imaging modalities, in particular diffusion imaging, which I mentioned earlier is a critical component of these microstructure imaging techniques. That is a substantially enhanced capability. So it allows us to get much, first of all, higher resolution. This is a Connecton scanner image. This is a kind of corresponding image from a standard hospital scanner, at least at the time. This was a while ago now. You can see that there's kind of a quantum leap in both image resolution, but there also is in contrast, sort of less easy to appreciate on the slide, which is amazing. But of course, this is just a one-off scanner. There's no way these are going to be deployed in every hospital anytime soon. As the power consumption of a nuclear submarine needs its own substation, costs a fortune. Siemens don't want to build lots of them. So yeah, it has sort of limited clinical applicability because there's only one of them. But it led me to ask the question, well, could we, from images that we get from a standard hospital scanner, learn a mapping to estimate the image that we would have got if we'd taken that patient, shifted them to Cardiff where we installed this super scanner and imaged them on the Connecton machine. And that's what image quality transfer aims to do, transfer the quality of this high, super image to the lower quality one, but more available. And of course, that idea adapts quite naturally to then estimating, say, a high field image from a low field scanner, and I'll come back to that a little bit later. So let me start with my sort of first attempt at this image quality transfer problem. Was very super simple. So simply get a set of matched pairs of low quality and high quality images, divide them into little patches, sample those patches kind of randomly from a collection of these paired low quality, high quality images, then learn a mapping from the low quality patches to the high quality patches. Once you've learned that mapping, you can then step through the low quality image patch by patch, apply the mapping to piece together a high quality image. So that the challenge then simply becomes learning that mapping from low quality patch to high quality patch. This was when I first started working on this 2013 or so. So it was pre-deep learning revolution really, and I just used an old school random forest to learn that mapping. But even that actually works surprisingly well. So here is just a simple qualitative example. This is a target high quality image and a sort of brutally downsampled version. We're going to try and estimate this high quality image from this low quality one. If you just do a simple interpolation, you get these kind of washed out, blurred out approximations to the high quality image. But even using this kind of simple random forest regression, patch regression, you actually you can get to a substantially better approximation to this this high quality image. You can see it a bit better looking at difference maps. See that the difference map between the enhanced low quality image and the target is much lower errors compared to the just interpolation. These are just various versions of the same thing on the right two columns. So yeah, not just pretty pictures, because so this is a nice slide showing that that enhancement actually has practical benefit. So one of the, I'm not sure how much people in the audience know about these different kinds of MRI, but one of the things you can do with diffusion MRI is to map the connectivity of the brain through a process of called tractography. So basically, you follow estimates of the fiber direction from point to point through the brain, and that gives you a reconstruction of the trajectory of that fiber pathway in the brain, joining different, they allow the different areas of the brain to talk to one another. So one of the challenges, one sort of common challenge for tractography that people have identified is these 4 distinct pathways in the brain that link the hand area of the motor cortex, it's up at the top of the head, to a region called the thalamus, which is a kind of junction box sending signals in different directions. And there are 4, you can sort of, anatomists dissecting the brain know that there are 4 distinct pathways between those 2 areas. But to recover those 4 pathways with tractography requires quite a high resolution image, and a higher resolution than one would normally be able to acquire on a standard hospital scanner, which would be more like this sort of 2.5 millimeter voxels rather than 1.25 millimeter. So here is just some tractography showing that we can recover those 4 pathways from a very high resolution image, and here showing that we can't recover those 4 pathways normally from a lower resolution 2.5 millimeter image. We just get a kind of a mess, really. But after the image quality transfer upsampling of that 2.5 millimeter resolution image, we can now nicely recover those four pathways. So, genuinely adds capability to those images for downstream processing. Another kind of nice result from that same paper, original IQT paper, showing really just a sort of tantalizing result, really showing that, if we say we learn a mapping from a 1.25 millimeter resolution image from 2.5 millimeter to 1.25 millimeter, can we assume some kind of fractal properties of brain images, which means that the mapping then from 1.25 to 0.63 is approximately the same. So we can apply that same mapping again, or to the original high resolution image to get a super high resolution image, and then even do it again to get an even higher resolution image. Tantalizing because it does produce some nice features. These tendrils of white matter in the cerebellum here, we know they exist and they kind of come into focus. These striations of the corpus callosum here, for those that know about the anatomy, again, we know they exist and they kind of come into focus, but of course kind of impossible to validate because we can't acquire images at this resolution. But still nice things that we can do with it. So later work on this technology, you won't be surprised to learn that deep learning substantially outperforms my old random forest implementation of this solution, and a huge amount of work went into designing the right kind of network architectures and so on for this problem from various students of mine, in particular Ryu Tano and Stefano Blumberg. And one of the key things that particularly Ryu did was to implement a sort of uncertainty estimates within these models, so that we could highlight, not just produce an enhanced image, but also a corresponding uncertainty map that would highlight regions of that image where the mapping is confident in the output that it's produced. It's seen similar things in the training data, for example, and other regions where it's less confident. For example, this kind of pathological region here is a tumor with a pathological surrounding to it that gets highlighted as high uncertainty. So that was the sort of entry point for image quality transfer, was looking at those images from the Connecton machine. But then shortly after that, I got some funding from the British government's Grand Global Challenges Research Fund to team up with a clinic in a town called Ibadan in Nigeria, to try and repurpose that technology for looking at images from low field MRI scanners. So in particular, their workhorse scanner, I hope you can just about see it through the scanner room window here, is this one. It's an open magnet, again, like I said earlier, sort of 1980s technology, really. Open fixed magnet. It's a 0.36 Tesla machine, and the question is that could we estimate from images acquired on this device the images that we would have got if we'd taken that same patient, flown them to London, stuck them in a modern 1.5T or 3T machine. So the kinds of image that were typically acquired on that device are quite different, actually, to the ones that we were looking at on the Connecton machine. They were no longer these diffusion images or quantitative maps, but much more basic MRI protocols looking at T1 weighted, T2 weighted, and flare images. And that required a little bit of redesign of the method. But here's just showing some contrasting some images that we get from the low field system with corresponding images from a high field system, and the output of an image quality transfer step to enhance the low field image. So I hope you can appreciate here that we get a substantial improvement in resolution in the through plane direction, and where the images from the low field system typically have fat slices, although quite high in plane resolution, and then in plane we also get a kind of an enhancement in the contrast. So you can see here a much clearer grey matter, white matter contrast. I won't dwell on the architecture, and it's basically under the hood is a fairly simple UNet architecture. One of the key things for getting this working is to accommodate the uncertainty in the mapping of intensities between the low field and the high field images. And so I think the key step in achieving the kind of result on this previous slide was constructing an augmented training data set that sort of accurately represented the variation in contrast mapping between low field and high field images. And just this is a kind of example from a clinical case where there's some quite difficult to detect epileptogenic lesions in the brain here. This is the low field image, but after the IQT enhancement, those lesions become much more conspicuous. And this is representative of the primary clinical application we had in mind for this technology, which is looking at pediatric epilepsy. The aim being to try and enhance the conspicuity of subtle lesions to enable better clinical decisions on what treatment approach to take. So the study on that continues now. We are acquiring from around 100 pediatric epilepsy patients, both low field images in Ibadan, and we then, once we've done that, we then bus those individuals down to the capital. It's not a capital anymore, is it? But down to Lagos, where there's a private clinic that has a 1.5T machine. So we get the equivalent high quality images from them there. And the aim of the study is to evaluate the impact of the image quality transfer on the clinical decision making. So it involves showing 3 images to a set of radiologists, the original low field, enhanced low field, and high field. And the hypothesis is that the reports from the enhanced low field approximate the reporting on the high field better than the low field. So some early results from that study just showing that basically sort of confirms that hypothesis so far. This is just getting the radiologists to assess the image quality in general, and you can see that the enhanced images in purple approximate the high field images in red, at least for T1 and T2, not so much for the flare, where we had some problems in the training that we're still working on. And there are even some radiological tasks where the enhanced images outperform even the high field images, which is interesting. And that's typically delineation of structures that require radiologists to look carefully at the out of plane images, so looking at the coronal or sagittal slices. So yeah, I mean, that a very simple study really, this work in Nigeria. But let me finish just that topic with some reflections on it. So one of the key things about running a project like that in quite an unfamiliar environment is getting the logistics right. It's a major challenge. How do you get the right permissions for busing people from Ibadan down to Lagos? What insurance do you need? What computers do you need in the hospital to implement this, and how do you get them there? How do you organize the data transfer? All of these things are basically impossible to do externally, so rely very heavily on having a very dedicated local team, and we've been lucky enough to have that, but who you have to put absolute trust in to enable these different aspects. The other one, another point, second kind of bullet point here, is that my immediate impression on the first time I went to Nigeria to visit the hospital we were working with was what a luxury item it is that we are trying to implement. Our sort of can we really justify all this effort and expense on a fancy, and not even that fancy, fairly simple kind of AI technique when the hospital is lacking internet and computers and even running water in certain places. And it is a challenging question, but the more that project has continued, the more I've seen the excitement that it generates. People being able to get involved in some of these, quite a high-tech project. It sparks a huge amount of interest, and it gives the sort of bigger activities like the camera network I mentioned earlier ammunition to then go and lobby the powers that be to work on that basic infrastructure to enable more of the kinds of example that we've been able to generate. So I do believe that it has value despite that sort of initial contradiction. Then the other thing that struck me, now the project is sort of pretty much in its final phases, is that the immediate impact doesn't come from where you initially expect. I still have some faith in the technology itself. I still believe that it offers some benefit, but by far the biggest impact of the project is not that. It's much more the impact it had on the patients who were part of the study. They get way better care than they would have got otherwise. And then there's the kind of knowledge transfer just from bringing for example, the radiologists in Nigeria together with those in the UK. There was a huge amount of transfer of knowledge in both directions. And then, the training opportunities it spun out, it spun out a lot of projects, student projects, both of medical students and computer science students in Ibadan, who we've, been lucky enough to work with. They're all super excited about, developing new AI tools and have, great capability to do so. So those things I think have been really the primary and most immediate impacts. So I guess the lesson there is doesn't necessarily come from where you initially expect. So let me finish just by talking about a newer project along similar lines in some ways. This is a much broader project. It is, for those that know about the Wellcome Leap, it's one of these grand challenges of the LEAP pose. And it's one of the first ones they put forward, actually, which was about understanding the brain development in the first 1,000 days. So looking at first 3 years of life. The project I'm involved in is called Kula. It's led by the wonderful Kirsty Donald here from Cape Town in South Africa. And the aim is to try and find ways to predict or predict the development of executive function over those first 3 years of life. It's basically a big data collection effort. We are looking at cohorts in various locations in Africa and acquiring various kinds of data, microbiome data, sleep data, EEG, and then there is an MRI component. So my little corner of this huge project is trying to help. This is the machine that we looked at earlier, the portable MRI machine from Hyperfine, is trying to help identify imaging biomarkers derived from low field images from this machine that are helpful in that task of predicting executive function over the first 3 years of life, and in particular detecting individuals in which that development is perhaps slow and could be accelerated by various interventions. So that really sort of there are two key demands. One is taking the pretty low quality images that come from these devices and deriving some simple numbers that can be used, put into a predictive model. So, simple things like brain volume would be one, but then we'd like to get a bit more fine-grained than that and look at maybe the volumes of different brain regions. That's a relatively simple task. Also in the task, much more challenging, is going back to some of my earlier slides, trying to get some measures of actual connectivity recovered from diffusion imaging and tractography. major challenge on a machine like this, but we'll come back to that in a minute. Slight rewind. This is another kind of technique that I was involved in developing, led by Eugenio Iglesias here. The technique called SynthSR, quite similar to image quality transfer, but addresses a slightly different problem. The problem that Eugenio set out to solve was, given any sort of grungy old clinical MRI scan, can we map that to the best guess high resolution MP-RAGE T1 weighted image? The reason you might want to do that is that if you get a nice high quality MP-RAGE, it will pass seamlessly through various image processing pipelines, such as FreeSurfer, to generate the brain volumes that I referred to on the previous slide. So that's the aim of SynthSR, but basically under the hood is quite a similar idea to image quality transfer, and works in a similar way. One very cool thing that SynthSR does is a bit like the data augmentation I mentioned with the low field to high field IQT, where we had to capture this uncertainty in the image contrast. SynthSR takes this to the extreme and augments sort of pairs of images in a very extreme way by completely randomizing the contrast. The aim being to learn the basic morphology of those images to do so that you can then segment different structures rather than having reliance on any particular kind of MR contrast. So that, very in a nutshell, is how the method works. It works surprisingly well even on the very low quality hyperfine images. So here is a typical hyperfine image. This is actually from an adult rather than a child, but going through that SynthSR enhancement, actually produces a pretty dramatic enhancement, and not a bad approximation to a reasonably high quality T1 weighted image. So Eugenio's just published a study showing a reasonable correlation of volumetrics, regional volumes derived from those enhanced images with what you get from the same people imaged on a high field system. It's not perfect, particularly in some of the scenarios that we're interested in, where we're not just interested in deriving biomarkers, but also want to produce higher quality images that can be reviewed by a radiologist. One of the things that SynthSR does is to obliterate lesions, like this one here, quite a clear lesion, gets kind of wiped out by the SynthSR process. SynthSR does that on purpose because then it makes it easier for the image to go through the image processing pipeline. But that's not ideal for some of the scenarios we're interested in. So one of the things we're working on in the Cooler project is to combine the original IQT, which sticks much more faithfully to the original data, including pathologies, with some of the capabilities of SynthSR. So now this is looking at some data from kids. This is a data set from a 3-year-old subject. Actually, morphologically, the brain of a 3-year-old doesn't look that different to an adult. So even with a model trained on adult data, we can do a reasonable enhancement and get a reasonable segmentation of the brain into anatomical regions and derive some nice biomarkers. It doesn't always work. Here is a case where the image is a bit grungier and the enhancement fails and then the segmentation fails. But I think, we are, as I say, we're still using models, early days in this project, and we're still using models that were trained on adults. And I think once we retrain that using more representative data, we should get a more robust system of enhancement and then segmentation. So that was the good news. The bad news is that the people that we really want to be able to image on this hyperfine system and derive markers from are the 3-month-olds, the infants, so that we can then make predictions about how they're going to progress over the next 3 years. The data from the infants on the hyperfine system is much, much grungier, barely any content at all to the T1 weighted images. T2s aren't too bad, at least some anatomy visible there, but they are not of sufficient quality at the moment for these enhancement algorithms to get their teeth into and produce a decent output. So that's really, a point for, there are some challenges there. First of all, in the imaging, which we have to work quite hard with to get better contrast here, and then in the enhancement by finding more representative training data. Nearly finished, in case you're wondering. So that, yeah, challenge number one, improve the infant imaging, then retraining the IQT to work better on data from kids. And then, of course, the last thing I mentioned when I started talking about this project was, can it really be possible to do, advanced imaging like diffusion imaging and tractography on these kinds of systems? Well, just to give you a taste, so part of the problem is that each single diffusion weighted image on a hyperfine system requires about 10 minutes of acquisition time. And the absolute minimum you need to do tractography is 7 of those images. So right there, there's an hour and 10 minutes minimum of acquisition time in order to even have a hope of doing diffusion tensor imaging, and that is just not feasible on kids. So what we're thinking about is ways of possibly speeding that up. And here is a courtesy of Derek Jones, who's part of the same project, just an early exemplar to at least prove that this is vaguely possible. So here is a 7-image acquisition. This is actually from an adult rather than a child, but nevertheless produces a reasonable diffusion image, an anisotropy map, and even the first example of tractography done on one of these hyperfine systems. Pretty basic, just picks out a couple of the main tracks. But actually, if we wind back the clock 20 years and look at the kind of tractography, this is when tractography first started appearing, and look at the tractography that people were doing then on high field systems, actually they were looking at just the same kinds of pathways, the corpus callosum and the corticospinal tracts, exactly the ones that we pick out here from the hyperfine. So, I think there's hope that over the next 10, 20 years, we can enhance this to the point where we can actually get some meaningful connectivity metrics, even from these very low power systems. So just to conclude, I think, there are some major innovations in accessible MRI going on at the moment, much of it motivated by this idea of democratization of MRI and deployment into a much wider proportion of the world, in particular to lower middle income settings. And AI is an absolutely crucial role in realizing that. The hyperfine system right now has a huge amount of machine learning and AI tools under the hood, not just for doing the kinds of enhancement that I talked about, but even just for reconstructing the images in the first place. But there are major opportunities there. It's done in quite a simplistic way at the moment, and so major opportunities to improve that and integrate it with the kinds of learning step that I've been talking about, where you sort of learn from external data. So yeah, lots of interesting things to do, but of course it's only a very small part of this overall challenge of fundamentally changing the infrastructure and educational sort of policies across some of these places that can enable them to take advantage of these advances as they happen. I shall stop there and thank many collaborators who helped with this work, people that gave me money, obligatory advert. We're always hiring. If any of these sound interested, do get in touch. And yes, thank you for listening.
Great. Daniel, thank you very much for that. That was terrific. So for the audience then, please post your questions in the chat and maybe I could kick off with one. Well, there's one question here from Jürgen Ivanis. Did the World Bank supporting the MRI availability in low-income countries so far as promised a few years ago so that people around the world have similar access to oncology prevention or treatment? I guess the question was perhaps policy one. Do you know what the investment landscape looks like, Daniel?
So from the World Bank, not brilliant as far as I'm aware. So I'm just trying to bring back the. I can see. There you are. Okay, got you. Where investment is coming from at the moment is many of the sort of charitable foundations. So people like the Bill and Melinda Gates Foundation, the Chan Zuckerberg Initiative, are putting quite a lot of money into the deployment of the kinds of new devices that I've been talking about into Africa and some of the South America, Southeast Asia, some of those lower income settings. So they are they're really putting their money where their mouths are, actually, and providing quite a lot of funding to support these initiatives.
Great. So Daniel, maybe I could ask you a kind of more operational question around the image transfer that you were describing before. So it seems to me that there are two kind of fundamental hard problems. One is that presumably there is pathology that is just not visible on the lower the lower magnet strength, and conversely, as you illustrated, I think in one of your later slides, the machine learning algorithm can almost wipe out the pathology because it's trying to map to the kind of trained map, right? How do you see that? How do you see getting around those issues then?
Yeah, it's a key challenge, obviously, because there are obvious dangers. That you hallucinate things or you obliterate things. Hallucinate things that aren't there or obliterate things that are there. And so that was one of the main things that motivated. I showed those uncertainty maps at some point, and that was one of the primary motivations behind that. I think there's actually quite a lot you can do just in the design of the solution to avoid a lot of those problems. I think that sort of obliteration and hallucination is much more of a problem if you try and estimate an entire brain image all in one go. Whereas I think, actually my sort of old initial paradigm with using the little patches is much less susceptible to that sort of problem. And I'm not saying it never does anything weird, because it does do weird things, but it won't do it will respect the original data much more than an algorithm that tries to construct an entire image all at once. So I know it's not a complete answer, but I do think that by sort of bringing the problem down to the more sort of micro or meso scale rather than the macroscopic scale of trying to estimate an entire brain image, you can actually circumvent some of those problems. But proving that is quite hard. How do you go about proving that a lot of the features and things that come into focus as you enhance an image are genuinely there?
And I guess a clinically important information, right, which comes back to your study in Nigeria. Can I ask you a kind of another question, which is around the training set size? Have you reached the limit? Would more mean better? What is the kind of optimal eventual performance of your algorithm given like loads more training?
So interestingly, again, that discussion of sort of patch and patch size versus entire image is quite salient to that as well, because the original work I did with the fairly small patches, they were typically 5 by 5 by 5 image patches that I put into the random forest that I described. Actually, the amount of training data you need to do that is pretty small. You reach kind of diminishing returns, with more than about 4 or 5 images, because I think there the problem's quite simple. Because you're looking at such small patches, there's no notion really of trying to learn anything about the sort of macroscopic morphology of the brain. It's more just about what does a little patch of image at this low resolution, what's it most likely to look like at high resolution? And actually, I think that mapping is quite simple, and you don't really need a huge number of examples to learn it. Plus the fact that if you have 4 or 5 images, you actually get quite a large stack of patches that are 5 by 5 by 5. The deep learning methods that I talked about later on sort of gradually start to use bigger and bigger patches, culminating in the SynthSR technique, which I described, which does actually work at the whole image scale. And I think the sort of simple answer to your question is that the bigger those patches are, the more data you need, because you get less patches per image, and the diversity of them becomes greater and greater and greater. But interestingly, you know, for the particularly for even sort of reasonably large patches, you know, say 11 by 11 that we used in some of the sort of early deep learning versions, you reach diminishing returns with a, you know, fairly modest data set, you know, certainly less than 100 images.
Does it output what features it's paying attention to? Would that be interesting in any way or not?
So you sort of can do that, but then, you know, when it's when you're looking at these, the patches themselves are already not particularly meaningful to look at in isolation, and so that, you know, the features that they latch onto are also not easy to interpret.
Helpful. Yeah.
Yeah.
Great. And Daniel, maybe I could ask you a question, because I was intrigued by your kind of different disease trajectories.
Oh yeah, yeah.
And does your approach allow for people to jump between trajectories, or is the assumption that someone's on a invariant path and that is different from another one? I mean,
yeah, it's a good question. And in fact, we've played with various kinds of models. So the one that I showed you does sort of make that assumption that, you know, people are assigned to a particular trajectory. I mean, that assignment's probabilistic. So some individuals, you know, very strongly identify with a particular trajectory, whereas there are others who, you know, I've got some other diagrams I didn't show. You get these sort of little triangles where you can place people. If they're on the corner, they're very strongly aligned with one subtype, whereas if they're in the middle of the triangle, you know, they're sort of equally likely to follow any of them. And you do see people clustering around the corners, but you certainly see some individuals who don't. Mm-hmm. And so that particular model works that way. You know, there's an assumption that people align with one of the subtypes and not others. But there are other kind of models a bit similar where, you know, you almost search instead for a kind of you know, a basis of trajectories, and so every individual then executes some combination of that basis. Yeah. Which is so you have to construct the model in a slightly different way if you work with that assumption, but in some ways, you know, has different advantages, I guess.
And the and in the case that you showed of the neurodegeneration, were the features that characterized people in certain groups helpful kind of biologically or medically? I mean, did they provide insight in any way? Or
I hope so, but so I mean, I think you know, definitely the biggest what's attracted attention to that work is the more pragmatic ability to stratify. Yeah. But you know, that was kind of where I started, really, was, you know, that we could get more insight into the biology by identifying these different subtypes, because, you know, staring at a kind of heterogeneous mass of people, it's very difficult to derive any conclusions about what might be the, you know, driving mechanisms that, you know, that cause the disease to progress that way. Mm-hmm. So I up to now, I think that we haven't quite probed that as much as we might have done, but it's something I'm thinking about quite hard now. Is you know, if you can, various people have constructed sort of computational models of how, you know, the pathology in, say, Alzheimer's disease spreads over the brain, and it's difficult to verify or sort of compare one computational model of the disease spread against another, against data when that data itself is heterogeneous, and there it might contain different subgroups, all of whom are behaving under different mechanisms. But if you can then divide people into more heterogeneous groups, then that gives you a better kind of starting point for evaluating the likelihood of various different mechanistic hypotheses. And so that would, there's a sort of,
or even selecting people for clinical trials, right? I mean, yeah, brilliant. Yeah. Well, look, Daniel, thank you very much. It looks like there's just messages saying thank you for the talk, which will be available on the YouTube channel next week. So thank you again, Daniel, for a terrific talk, and thanks everyone for joining.
Great pleasure. Thanks for having me.
Cheers. Bye.
See you.